Today, let us start with cell structure. Histology. Histology is study of tissue, cells, and organs. We use light microscope to magnify the substances like tissues and cells, and it magnifies up to 1500 times. Now, cell. Here you can see cell is a structure which has basic components of cell are cell membrane is also known as plasma membrane, then cytoplasm where it contains fluid like matrix like substance, and nucleus, and here we have membrane bounded organelles. They're also known as vacuoloplasm. Now here cytoplasm with nucleoplasm. Inside the nucleus we have nucleoplasm, so cytoplasm plus nucleoplasm is also known as protoplasm. Now let us move on to the cell membrane. Cell membrane is made up of phospholipids and associated proteins, and it is also known as plasma membrane. Now, cell membrane is about 7.5 nanometer thick, and it shows light and dark staining. So, so that it appears like this, and it appears like trilaminar layer, but it is actually lipid bilayer. And most of the lipid bilayer is made up of 50% is made up of lipids, and 47% is made up of protein, and only 3% is made up of carbohydrates. Now here we will take one phospholipid from here. Phospholipid, it has head as well as tails. It has one head and two tails. The first head is phosphate portion. It is what and this is water soluble, so we'll call it as hydrophilic, and it has some charge, so it is polar, and it strains when we strain this part we'll get dark strain, so it is dark staining. And this is lipid portion, which is hydrophobic, that is it is lipid soluble, not water soluble, and non-polar, it doesn't have any charge. Then it shows light staining when we stain it. And now here we can see the phospholipid is again of two types, saturated as well as non-saturated, and unsaturated or non-saturated. Now here the lipid bilayer, we can see that they are arranged like this. This is the outer phospholipid, then this is the inner phospholipid. The outer phospholipid and inner phospholipid are asymmetrical in nature, because the outer phospholipid can bind to carbohydrates, etc. And here you can see the layer. This is called as glycocalyx or cell coat, which is made up of carbohydrates. And now this upper part is made up of phosphatidylcholine and sphygmomyelin, where myelin consists of major compound known as galactocerebroside. Next, the lower part is made up of phosphatidyl and phosphatidylserine. Now this is the bilayer, lipid bilayer, which is seen in the cell membrane or plasma membrane. Now here you can see this. Sometimes we see cholesterol between the phospholipids, which have a hydroxyl group at the end to show, because it is polar in nature. Now phospholipids, here you can see it is flowing. It is flowing like diagram. So we can say by this the phospholipids show fluidity. For example, we can see this fluidity in endocytosis, exocytosis. We'll know that later. And fluidity is important in biogenesis. When temperature increases, the fluidity decreases. Sorry, when temperature increases, the fluidity increases. And when cholesterol, here you can see when cholesterol is increasing, the fluidity decreases, because here you can see it will become congested between those, so it cannot move easily. So the fluidity decreases. Now here, as I said, the phospholipid is of two types: saturated, non-saturated, or unsaturated. Here, if unsaturated lipids are more, the fluidity will increase. Sometimes saturated lipids will form tightly packed structure with cholesterol between them like this. So here receptor protein is a protein, carrier protein, which helps in situation like this, like tightly packed protein tight packed lipid layer is present, then receptor protein will help us. This receptor protein is also known as 7 pass receptor, because it is passing throughout the lipid membrane 7 times, 1 2 3 4 5 7. Then it has an associated protein, and this is receptor where the molecule is binded, and the stimuli is taken from external stimuli, and it is giving signals inside the cell, so it is converting external stimuli to internal stimuli, where we can say receptor with associated protein is also known as lipid raft. And the lipid layer is fat soluble, so only small molecules and lipid soluble molecules can only pass through the lipid layer, whereas polar molecules or non-lipid layer water soluble molecules, they cannot pass through the cell membrane. So we need different modes of transportation. We'll see different modes of transportation. First one is also known as simple diffusion. It moves from high concentration to low concentration, and it is applicable to small and lipid soluble substances, because high it does not involve any proteins or carriers. See here, it is passive in nature, it doesn't require any energy or ATP, whereas it doesn't have any carrier proteins, so only small particles and lipid soluble substances can move from the with the help of simple diffusion. For example, here you can see oxygen is moving from outside of the cell to inside the cell, while the concentration of oxygen is more outside the cell, whereas it is less in inside, so it is moving through diffusion. Similarly, the concentration of carbon dioxide is more inside the cell, and it is less inside the outside the cell, so carbon dioxide is moving from inside cell to outside cell. Now here simple diffusion is proportional to the concentration gradient, surface area, then solubility, lipid solubility, and it is inversely proportional to thickness of the membrane and size, molecular weight of the membrane. It is passive and no carrier proteins are present in this simple diffusion. Now let us see, before going to the second one, let us see about types of protein. We have two types of proteins, that is integral and peripheral proteins. Now first let us see about integral. Here in the diagram you can see this is a membrane, this is a membrane, and the integral proteins are throughout the membrane, that is above, and they are passing throughout the membrane, so they are known as integral proteins. And these integral proteins are of four types. Carrier proteins, that is they take the material or molecule from outside of the cell and they carry it inside the cell. They are present throughout the membrane, so they come under integral proteins. Then enzyme is a substance where it converts it may be, here also you can see the enzymes, it may be peripheral as well as integral. Both the functions are same in peripheral as well as proteins. Enzyme converts one molecule into another. For example, if we take the enzyme came with A plus B, then it releases C plus D. Next is linker protein. Linker protein helps in connection of intercytoskeleton to external cell environment. Then receptor proteins, we already studied about receptor proteins. Then next, peripheral proteins, they are only present on the surface, they are not true, they may be on upper surface or lower surface, but they are on the surface, not throughout the membrane. There are of two types. Receptor associated protein, we already studied receptor proteins, in that we saw receptor associated proteins. Then enzymes. Now the second type of transport is also known as facilitated diffusion. Facilitated diffusion needs carrier proteins for transportation, and it does not need any ATP, and it moves from high concentration to low concentration. So it is moving according to diffusion, so it doesn't need any ATP or energy. Now channels are made up of um, here you can see it makes some channels like this, which are made up of peptides. which form here the space is left so that the water tunnel is made, so that the molecules from outside can go inside easily, which are water soluble or not non-polar or polar. So examples are sodium channel, calcium channel, and potassium leaky channel. Potassium leaky channel means this channel is always open. Potassium from the cell will always go outside with the help of this channel. Then sodium voltage channel. Here in sodium voltage channel, the voltage channel has a voltage sensitive gate which will only open according to the situation. Then ligand operative channel. This only open to the specific neurotransmitter or hormones. It doesn't open for every molecule. Now let us know about third transport system, which is known as active transport system. Now it uses active transport. In the name only you can say it moves from low concentration to high concentration, where it uses ATP and carrier proteins are also present. And it is divided into two types: primary ATP, primary active transport, as well as secondary active transport. Here in primary ATP or energy is directly used, whereas here ATP is indirectly used. Here you can see example is sodium potassium ATPase and calcium ATPase, and here sodium glucose and sodium amino acids. Here first example is sodium potassium ATPase. Here the 3 sodium are present and 2 potassium are present on this side. When they move from low concentration to high concentration, so we need energy. Here the special structure is formed known as ATPase, where ATP is taken and break down into ADP and phosphate. Now the breaked energy is directly used by this structure to flip this molecule. So we'll get 2 potassium this side, then 3 sodium this side. It will shift from here to here. And next is calcium ATPase, second example. Here calcium ATPase is done in the endoplasmic reticulum, where calcium is needed. So calcium from outside will go inside due to this receptor, where ATP is break down into ADP and phosphate and energy is used directly. So calcium will go inside the endoplasmic reticulum. Next is secondary or ATP individual. ATP will take indirectly. So we saw sodium and glucose is like this. It will use the energy from sodium and potassium ATPase. It will take ATP from here to here, and it will use it. So it is using ATP or energy indirectly. And here you can see 2 molecules are moving, sodium as well as glucose. If sodium is not there, then this carrier protein will not take glucose alone. So this type of transport is known as symport or co-transport. Here 2 are oppositely moving. This type of transport is known as antiport or counter transport. Now let us see about fourth transport system. Before fourth transport system, let us see this. Here the facilitated primary and secondary active transports are carrier mediated transport system, and they show some certain they show certain special features like stereospecificity. Like for example, if glucose channel is present, where we have two types of glucose like glucose L and glucose D, glucose levo and glucose dextrorotatory, but glucose channel only allows glucose D. It doesn't even allow glucose L. That is stereospecificity, which shows specificity. Each channel has its own specificity. Then saturation. If in the case of diabetes mellitus, we can see the saturation, where for example the insulin is not working properly, then the blood in the insulin will increase, and all the channels are busy, and the channels are over, they are occupied with the entry of glucose, so there is no more channels left. So we can see glucose in urine. This is known as saturation. Then competition, where here the similar molecules will compete with each other to pass through the channels. For example, glucose and galactose are similar in with structure and molecule, so they will compete with each other to go inside the channels. Now let us see about fourth system. Fourth system is also known as vesicular transport mechanism. It is of two types: endocytosis, exocytosis. In endocytosis, the cell will take material inside of it, and in exocytosis, it will remove the material from the cell to outside environment. And here in endocytosis, we have three types. Pinocytosis. Here, no specific molecule is taken, only water with its some soluble vitamins molecules are taken inside. Uh, and phagocytosis. Here, foreign materials are engulfed, like bacteria, fungi, virus, example, etc. And it takes helps of actin fibers to pull that material inside. Then retromediated mediated endocytosis. Here it takes the molecules with the help of clathrins. So today we have completed part 1 of cell structure. Tomorrow we'll see part 2. Thank you.